Share Human, share your screen.
Okay.
Hello everyone, welcome back to our e-seminar series for Translational Biomedical Engineering. It's with great pleasure to host Professor Mehdi Nikkhah from Arizona State University today, who will be talking about tissue-on-chip technologies for disease modeling. Before we start his talk, I'd like to go over a few housekeeping notes. First of all, if you have any questions during the talk, please write the question in the Ask a Question box. Also, there is a poll available for you to share your thoughts and feedback with us about this e-seminar series. Feel free to email me or Human via email. Send email to me and Human if you have any concerns, any suggestions, or any feedback regarding this seminar series. Next week, we will have Professor Ritu Raman from MIT, who will be talking about neuromuscular actuators for medicine and machines. Mark your calendars. It will be the same time next Wednesday. You can also follow us on Twitter using our Twitter handle TransLBME to get most up-to-date information about this e-seminar series. Last but not least, I would like to thank our sponsor, Montreal TransMedTech Institute, who has been with us since almost the beginning of this seminar series. Thank you, Montreal TransMedTech, for all your support. So with that, I'd like to introduce our speaker today, Professor Mehdi Nikkhah, who is currently an associate professor of biomedical engineering at the School of Biological and Health Systems Engineering at the at Arizona State University. Dr. Nikkhah completed his postdoctoral fellowship training at Harvard Medical School and Brigham and Women's Hospital. He received his bachelor's degree in mechanical engineering and a master's degree in biomedical engineering from Tehran Polytechnic University, followed by a PhD degree in mechanical engineering from Virginia Tech. His lab's research interest is focused on the integration of micro and nanoscale technologies, innovative biomaterials, and biology to better understand the mechanisms of human disease progression and also to develop regenerative medicine strategies for treatment of organ or tissue failure. Dr. Nikkhah has published more than 65 journal articles, 8 book chapters, and 85 peer-reviewed conference papers, and holds 4 US patents with more than 7,000 citations and an H-index of 40. He has received numerous prestigious awards and recognitions during his career, including Arizona Flynn Foundation Award, Arizona State University Biomedical Engineering Outstanding Assistant Professor Award, NSF Career Award, and so forth. Along with his research endeavors, Dr. Nikkhah has been very passionate to educate the next generation of students and scholars with specific attention to minority and underrepresented groups in science and engineering. He has trained more than 45 postdoctoral fellows, PhD and master students, as well as undergraduate students from a diverse background in his lab. With that, I would like to thank Professor Nikkhah for accepting our invitation to give a talk in this e-seminar series. Welcome, and the virtual floor is now yours.
Thank you very much to the hosts, Mohsen and Human, for your kind invitation and introduction, actually. And then it's a pleasure to be here today for your e-seminar series. And I would like to acknowledge and mention that this is a great initiative by you to organize this series and fantastic seminars to bring the community together, especially during this COVID situation when there have been social distancing, people have been apart. These are great learnings. And the other thing I just want to mention about this seminar series is like it transcends the boundaries in my eye. Like it doesn't need any person, you don't need to be in any specific location to attend these seminars. Like students from all over the world and scholars can attend these seminars and provides equal opportunity for everyone. And that's what matters at the end of the day, that we need to provide. That's our job as educators. And then you guys have done a fantastic job. So congratulations to you guys. So should I share my screen now? I think let me go ahead and share my screen. If you do you see my slides?
Yes, yes. Thank you.
Excellent. So I think if I get disconnected, please let me know. But I
Sure.
hope it kind of works smoothly. So as Mohsen mentioned, my name is Mehdi Nikkhah. I'm associate professor at Arizona State University Biomedical Engineering. So I have been at ASU since 2014. And then I started as assistant professor and then was promoted to associate level last year. So in the talk today, I would like to discuss and present some of our work on organ-on-chip and tissue-on-chip technologies, specifically for disease modeling applications. But because of the hosts asked me to talk a little bit about my background, educational background, especially for students, and where I'm coming from, I would like to dedicate a few slides, like 3 to 4 slides, about where I'm coming from, and then where did I start my education? So I was born in Iran, and these are the slides that I share in my ASU 101 class, and this is ASU BME 101. That these are to the freshman students, and we have to tell the same story and that where we are coming from in terms of science, and then where we started our education. So I started basically my education at Amir Kabir University of Technology or Tehran Polytechnic with a bachelor's in mechanical engineering and then master's in biomedical engineering. But believe it or not, when I got admitted to mechanical engineering in year 2 and 3, I realized that I don't want to be a mechanical engineer by the end of basically my career. And then I didn't like that much no offense, but portion of my major with respect to machine design and those kind of things. But it was fascinating. But then I finished, and then I got admitted to biomedical engineering, which was one of the good incidents which happened to me, which was a little bit unplanned. I mean, I selected mechanical engineering to go into master's degree, but then I got admitted to biomedical engineering. For those of you who know how the major selection is in Iran, actually. So, but it was a good happening, and I was not aware of it until the future years came, and I realized I have been blessed to be admitted into this major. And then, and that was the time that I decided to immigrate and then continue my education to a higher level. And I showed this sign on my next school when I moved. And then I tell students here at ASU and say, can you guys guess where this school is located? And it's like suburbs of Philadelphia. They have a very good basketball team. And then, so I was admitted to a school. It was a fairly small school, 8,000 students, called Villanova University. And then they didn't have a biomedical engineering, they had mechanical engineering. But when I started my biomedical engineering degree at Amir Kabir or Tehran Polytechnic, my main focus was on human gait and walking, and then locomotion, basically. And then at Villanova University, I continued on the same research, but it was under the umbrella of bioengineering, but in totally different area of rehabilitation and locomotion. And believe it or not, this was the first paper that I published. And then I hope you guys see my screen because I don't see you guys. But so robust control of under-actuated bipeds using sliding mode controls. And then what you see, this is the first paper published is totally in a different area in robotics. And then what you see on the right picture, excuse me, do you see my mouse when I move my cursor?
Can you move it?
Yeah, I move it now. Do you see that or no? You don't see that?
Yes, I see.
Yes, I can see it.
So I just want to point. So this was actually a biped, meaning like it was a humanoid robot which was walking and for 120 steps, and it was a totally different area. So that was the time when I finished my master's, and I got a little bit tired of math equations, and then so these systems requires a lot of mathematical formulation. It's under the umbrella of control theory, and you have to write equations, and it was fascinating, and a lot of coding and MATLAB coding to let this robot walk and do not fall, and it was interesting, but it was not. I decided to change my major actually, and then like under the same category of bioengineering, but in a different research area. So that was the time that I was admitted to Virginia Tech for my PhD in mechanical engineering, and there was a professor, Masood Agha, as you see on this slide, and his photo is here. And then this is the photo I took from their lab website called VT MEMS Lab, and they have still my one of my photos here when I was a graduate student presenting a poster in a conference. So he was an assistant professor there during that time. He started his lab in 2005, and I joined Virginia Tech in 2006, 1 year after he started his lab. And then he was an expert in MEMS and microelectromechanical systems, especially for gas chromatography. And then he mentioned that he would like to start his bio lab. And then, so it was a good integration of skills and the merging of skills that I joined this lab and then continued my PhD. But my research mainly was totally became different. And then I started working on generating silicon-based microtopographies or microscale textured features, as you see in these photos, and then to study how cancer cells behave on these microtopographies and how we can distinguish the biomechanical signatures of cancer cells from normal cells, and whether there is a hope that we can use these platforms, as you see, as a biosensor, a microsensor for isolation of tumor cells in a population of normal cells. So what you see, actually, this is the chip, and then you see the coin here, and then this is a single fibroblast cells on this SCM image sitting inside this topography, and then bottom actually shows actually a movie of cancer cell capturing. So this was the first time that I was introduced to the field of cancer. And then there was a lady, Dr. Janine Strobel. She was a pharmacologist. She taught me all the cell culture biological assays, and it was a totally different research area. But it was very fruitful. I would call it the golden years of my education. Then I decided to continue on my PhD, my postdoctoral fellowship. So I got admitted, and I was privileged basically to go to Harvard Medical School and work with Professor Ali Qadem Hosseini. He was my mentor, as you see here. I think this photo was taken, I believe, in 2013 on the top photo, and then Mohsen is here also, that is the host here. And so it was a large group, and it was I was privileged to basically to be mentored by him, and I learned in his lab, in Ali's lab, about hydrogels and biomaterials, and especially tissue engineering with specific focus on cardiovascular tissue engineering. And the area was like, how could we build microscale tissue models to generate vascularized constructs for regeneration of myocardial ischemia, or basically heart disease, and etc. So this was the introduction step to cardiovascular tissue engineering. Now, then after this period, which was a very also very fruitful, and then great years, to have great friends, and basically I got to know so many great people from this group that we are still colleagues. And then and in the field, Mohsen is one of them, as I mentioned, and then I got to know a lot of other people that they have presented also in this seminar series, in the past seminars. So with that, I came to Arizona State University in 2014, January of 2014, and I started my lab. So this is the umbrella of the lab, and I have this slide to show basically where our research landscape is focused. So we are work at the interface of micro and nanoscale engineering, and then biomaterials, or hydrogel-based biomaterials mainly, and then biology and medicine, toward two major areas. One is regenerative medicine and tissue engineering, and one is tissue on chip technologies or disease modeling. So when I started my lab, I tried to merge the two areas of what I learned during my PhD training and postdoc trainings, and open new basically initiative or create new initiatives. And then one of my passion was to develop tissue engineered models of tumor microenvironment or cancer. And that was something I wanted to follow and I pursue, and I will share some of the projects that we have done during the years here. So today I would like to talk a little bit about the technologies that we have developed actually in our lab for cancer research and tumor microenvironment. So I have a few slides, like 3-4 slides about OrganonChip in general, and then I'll jump into the research and I explain and I'll show you basically some of the projects that we have been working on. So as most of you are aware, the process of drug development in pharma is a very complex, arduous, lengthy, costly, and then complicated process. And it requires a lot of basically steps, from basically standard assay to preclinical studies, animal studies, all the way to the patients. Now, it takes about $2.5 billion and 10 to 12 years to develop one compound that the patients actually use. So that you see that cost and the years are very basically lengthy. And then a lot of drug candidates which come to the initial phase of investigation, they actually fail in the preclinical models or animal models that they use. And mainly the animal models that are being used for pharma or in drug screening assays or drug development are basically, for instance, rodents. But the animal models, there are severe limitation or major limitation with those. First, they're not humans, so the cells are not basically human. So you can create tumor models or entities of tissues, PDX model using human cells, but the whole organ and the whole basically entity is not human, and the surrounding microenvironment especially are not basically human cells. And then the complexities in the physiology. So if we want to dissect a specific factor in an animal model and to see, for instance, what kind of specific immune cells or stromal cells promote tumor invasion that I'm going to talk about, we cannot dissect the influence of that specific cell types because of so many confounding factors. And also the other challenge is the imaging and high resolution imaging. So there's a notion in the drug discovery, they say fail early and fail cheaply. So it's better to fail early and know that that compound does not work. But there's also an issue with that. A lot of compounds that they have failed in the early process, actually, if you continue that to the later process, it may have worked in the human. So they're being eliminated because in these assays, they didn't show efficacy. So there is a need to develop predictive and complex models which are utilized in human cells and recapitulating the complexities of human physiology, and they have the power of preclinical studies. So this photo was taken by a great review article written by our colleague Shuaik Zhang and also Melissa Radisik that they presented in this seminar series. So what they actually showed is OrganonChip, the conceptual figure for OrganonChip, that these are miniaturized or microscale platform technologies that they represent the whole organ function. And they could be created by human cells, as I mentioned. They could have basically vascular networks. We can control numerous microenvironmental factors such as stiffness, mechanical stimuli, etc. Now, what you see, the first paper, the name of OrganonChip, although the rich knowledge was there before 2010, the first paper, which was long on a chip, was published in Science, was 2010 by Dan Haw and Don Ingber's group. But the community has already developed and generated a rich knowledge on stem cell culture and tissue engineering, creating 3D tissue constructs that led to that point that the name of OrganonChip kind of like emerged, or I would call it the brand of OrganonChip emerged in 2010. But you see the number of cited papers which came out compared to microfluidics exponentially kind of surged all the way to 2016 and 17, which is the statistics is showing. And now the market for OrganonChip as of 2020, it was about $50 million, and it's expected to triple to 170, 280 versus $200 million in 2025, and it's going to grow further and further. But what this slide actually summarizes, and it was taken from this lab on a chip paper, summarizes companies and startup companies which have been developed in organ on chip areas. And then as you see, skim down through this, like we have liver on chip, and then we have vascular network on chip, tumor on chip, and then special cardiac on chip developed by different groups, and also lung on a chip. And then there are numerous startups in this area. It's a very competitive field, and then a lot of groups are every day developing different basically organ on chip technologies. But in my point of view, the merrier. Meaning, like, if we develop more platforms, and then there are more examples or there are more entities that we can compare the physiological relevance, and then we are going to get to a point that there are checkpoints that, okay, whether we are going to get closer and closer to the human physiology. So all of these platforms, they have a uniqueness in them that they can report a new finding. And that's basically the uniqueness and the beauty of this field. Although, as I mentioned, there's so many platforms, but it's very interesting. So it's a growing market. And then mainly most of these platforms, they had utilized microfluidics, but there are limitations in some of those. As we grow through the basically technology, the limitations are being addressed. So So what I want to talk to you today is about some of the projects in our lab. So as I mentioned, my interest has been specifically on tumor on chip and cancer, or tumor microenvironment on the chip, because of when I started, it was my training and also was my passion to develop tumor on chip. So I would like to talk a little bit about the cancer and show you some of the technologies that we have developed, and I show our other technologies on organ-on-chip area that we have engineered so far. So we know that cancer is the leading cause of death worldwide, and then 1.7 million new cases with 600,000 deaths. And then a major challenge in the treatment of the cancer is the complexity of the local tumor microenvironment. Okay, so what you see in this cartoon image, this is the local tumor microenvironment, which is comprised of multiple cell types, such as fibroblast cells, immune cells, epithelial cells, myoepithelial cells, and then tumor-associated macrophages, endothelial cells, which is the vasculature. And then when the tumor actually forms, actually the signaling between the tumor and the surrounding microenvironment, as you see, these are ECM fibers or extracellular matrix fibers. The signaling between these two lead to basically promotion of tumor cell invasion or metastasis into the vasculature, circulation, and then formation of a secondary tumor site, which all lead together to the patient's death. So this is a very, very complex disease landscape, okay? And then now, as I mentioned, if you want to use animal models, definitely there have been a great advantage into the use of animal models. So many investigators have used that so far to study cancer cell invasion or tumor cell metastasis. But because of the limitations, we would like to develop models which complement those animal models or address the limitations. And I would think of like the organ on chip or tumor on chip that we develop, it's going to definitely be complemented by animal models. So we are not at a stage to kind of fully eliminate the animal models for these studies. So bearing that in mind, we wanted to mimic these complexities on a microfluidic chip. So what you see, one of our first types of cancer that we started studying was breast cancer. So the breast cancer, the majority of types of breast cancer is actually emerged because of ductal carcinoma. So meaning that this is the duct. These are basically the tubes that they carry the milk. The epithelial cells on the inner lining of the duct, they go through the transformation to the invasive cells, and then they rupture the duct, they come out, and these green cells, as you see here, are fibroblast cells, and the macrophages are Purpur cells. What you see on the right image is the histology of the tumor versus the cartoon on the left, that it shows the tumor entity, which is in a circular duct, and the stroma cells around it. But interestingly, the initial architecture of this entity is very interesting. But later on, as time goes on, it becomes the intermingled with the tumor cells, with the stromal cells, the tumors. Now, what the question we wanted to ask was, like, can we replicate this architecture and then now go to fundamental biological studies? And I'm interested personally in technology and biology, so I'm not at the phase of like just technology development. But I would like to dive into the biology and then find the cause, which is very interesting, and I'll show you some of those results. So first criteria, if you want to design a chip for this specific entity, it should have an organotypic architecture with 3D cell environment, like with configurable 3D cells, so we can have cancer cells at the core, surrounding stromal cells. And now what is the matrix on the tumor cells are embedded? What is the matrix on the stroma? So there should be a spatial organization, and also how do we control the diffusion of biochemical cues or biochemical growth factors into the tumor from the mimicking the vasculature toward the tumor? And then how do we want to image the cells? Does our platform enable high resolution imaging of a single cell, and then whether we can measure a migration matrix? So this was the chip that we developed. So if you dissect basically the half of the tumor into two, you can see that symmetrical kind of feature. So we can use that half symmetry into the tumor chip. And then now we developed this microfluidic chip. I don't know if, so this is the chip that what you see on the left side is the tumor, 3D tumor surrounded by a stroma. And this is the microfluidic chip next to a dime. Sorry, next to a penny. And then, and then this is the micro. I don't know if you guys see that or not. Do you guys see this chip on the camera?
Yes.
Yeah,
yes.
This is the chip that I'm holding on my hand right now. And then Whenever I teach, I show these chips to students. So it's like just to have an idea. These chips, similar to other OrganonChip, most of OrganonChip platforms are fabricated in PDMS, which is the basically elastomeric polymer, polydimethyl siloxane. It's it's cross-linkable by heat or basically temperature, and has a cross-linking agent. You can shape it into the form that you would like. But the interesting part of this is like we created the 3D tumor on this red region, which is separated by a stroma by these posts. These are trapezoidal posts. And then now we can load cancer cells into any matrix that we want. We can switch the matrix on the stroma. We can add 2 cell types to the stroma, 1 cell type to the cancer cells, and we can place with so many factors here. And it has an organotypic architecture. We use COMSOL multi-physics simulation to look at the biochemical basic gradient of biochemical cues or factors into the tumor entity. Now, these actually the tumors that we have developed have led to several publications in numerous journals and has been granted 2 patents so far. And I'll show you, this was the 2-layer platform that was the first patent. But I don't know if you see the movie, I'm playing the movie. These are the cancer cells are crawling out actually at the single cell level into the stroma. So what we actually load cancer cells, we let it grow inside the matrigel for 3 days, and then these cells start invading. So this is the first qualitative image that we get. This is the high resolution of the tumor on chip. And what you see here, the size of the tumor is about around 1.5 millimeter, and the stroma is about 3 millimeters. But now the question comes, can we do single cell analysis? I don't know if I'm playing this movie. Hopefully it's gonna play on the screen as you see. But now the blocks that you see on these actually right movie, these are single cells. So we can actually see how every single cell is actually crawling in XYZ dimension. And this was the first study that we did. We can measure migration track of these single cells in response to epidermal growth factor. That was basically the factor that we used to validate the technology and show that these tumor cells invade. And this is the growth factor which are released by macrophages in the tumor microenvironment. So this was our first publication that we actually came with. And now going back to the tumor microenvironment, and then these are basically, this is a breast tumor microenvironment. Now these ducts or breast ducts that initiates the tumor initiation happens there is surrounded by extracellular matrix, green cells or fibroblast cells, and the purples or macrophages, as I mentioned. But now there's a complex signaling between these components, right? There we have immune cells, as I mentioned. So we just eliminate for a second endothelial cells, but now we have macrophages on the 3 axis with fibroblasts cells and the tumor cells, and they cross-talk to each other to promote the invasion of these tumor cells. So there have been so many studies using animal models to dissect how these macrophages, fibroblast cells kind of cooperate to promote cell invasion, because these fibroblast cells at the initiation are normal fibroblast cells, but because of signaling from tumor cells, they become diseased fibroblast cells or cancer-associated fibroblast cells, which all together promote tumor invasion. So it's a very complex kind of signaling. Now we are interested in these axes, like at the initially, like how these tumor cells signal to fibroblast cells or fibroblasts back to tumor cells, which promote the tumor cell invasion. And then, so what are the basically genetic factors or genetic instability induced by fibroblasts cells which promote the tumor invasion? So for this, we collaborated. This is where the translational aspect came a little bit. We collaborated on our second project with a fantastic surgical oncology physician, Dr. Barbara Pachka in Mayo Clinic of Arizona. So we isolated patient tumor tissues at Mayo Clinic in Arizona. So we would go with my student to the basically to Mayo Clinic. They would call us sometimes that we have a patient, and often getting these tumor tissues are very difficult. So it's not especially with patients such as triple negative breast cancer patients, it's very difficult. So it's like a huge kind of like demand for these tissues because people want to study actually use these tissues for themselves. So you would be called that there's a patient coming into the operating room. So, and then I remember one time we had to drive and then with my student, and then we actually collected the tissues from his pathology lab, and then the patient was in the OR. So we bring the tissues back to our lab at ASU, and then we isolate these fibroblast cells, and then now when we develop the tumor, so we have the tumors at the core, and then the fibroblast cells are surrounded the tumor into the stroma. So these are the 3 patients that we studied. First patient was 71 years old. It has an infiltrating lobular ER positive, PR positive cancer, and the second patient is 50 years old, ER positive, PR positive. But the third patient, if you pay attention on this third patient, she's a triple negative patient, and then this is one of the most basically aggressive types of breast cancer, which causes patient death. So we actually took these fibroblast cells and we put them in our tumor model, and then we have our tumor cells at the core on day 1 and day 3. What you see, actually, these are different conditions. I don't want to get into too much details, but the interesting part is, like, when we have these CAFs or cancer-associated fibroblast cells from these triple negative patients, it increases the invasion of tumor cells significantly as compared to these to the other type of fibroblast cells. And then this is the blue bar that you see here. And now, interestingly, the normal fibroblast cells, which are derived from bilateral mastectomy, meaning like they are derived from the other side, the other breast on the other side, which is a normal breast, they inhibit the invasion. So we don't know what is the cause of that yet, but that's a very interesting phenomenon to investigate. So we actually kind of specifically pinpointed on this CAF3, so we call it cancer-associated fibroblast 3 or CAF3 coming from patient 3. Now, we isolated cells from our platform, so I don't want to get into too much details here, but I just want to show you the capabilities of this platform. So most of the standard biological assays that you do, they're capable or compatible with these platforms. So we can extract the cells, sort the cells, and then you can do RNA sequencing, or we can do even single cell RNA sequencing. So that's what we are doing right now. But this was specifically RNA sequencing. We see that the co-culture conditions are clustered together, normal fibroblast clustered separately, and then when you have monoculture, is basically separated from the conditions. So that shows that there is a differential expressions of the gene when you have these conditions. Now, we had to go through we have collaborated with bioinformatics colleagues, and we had to go through multiple levels of statistical analysis. We found 280 differentially expressed genes because of these CAF3 versus normal condition, and out of those, through another level of statistical basically analysis, we found 20 genes, and across those 20 genes, what you see, do you see this box here on the mouse that I'm showing? This is called, we found this gene which is differentially expressed in control condition versus control and CAF, that when we have this cancer-associated fibroblasts from the triple negative patient. And then this called is glycoprotein non-metastatic B, which encodes the transmembrane glycoprotein in the cancer cells. So what it meant was, like, when we have this CAF from triple negative patients, and then we bring it to the close proximity of the tumor, this gene gets upregulated. Now, interestingly, my student Don, who performed this work, was my first PhD student, and then now he's in MD Anderson Cancer Research now. He digged into 2 clinical databases, basic TCGA, and then this is the other database, on 463 and 2,509 patients. And then what we found, interestingly, that the gene that we found in our platform was well correlated to clinical databases. What you see actually on the left, on the panel C, we see the GPNMB mRNA expression on the patients with carcinoma or cancer was highly expressed versus the control condition. And also the patients which had on the panel D, what you see, that means the patients which had higher expression of this gene, they died faster, actually. So there was a clinical correlation of what we found in our chip technology, and also like basically on clinical databases. Now, next step, we said we want to knock down these genes. So there is a lot of biology here. So we use shRNA knockdown to kind of knock down this GPNMB with specific gene, and we see the proteins are less expressed on the transmembrane on SHG1 and SHG4. And then we hypothesize that if we knock down that gene or silent that gene, basically the invasion of cancer cells, we can inhibit the invasion of tumor cells or cancer cells. So what you see here, the cancer cells on monoculture, we knock down, they're significantly less invasive. And then now when we actually add CAFs, so we can add the same patient fibroblast from the same patient to the proximity of the tumor cell and knock down the gene. And then we see the invasion is blunted or basically inhibited significantly. So this was an interesting finding for us, that testing our hypothesis, that it was actually that knocking down of this gene inhibits the invasion, and then it showed that this gene plays a significant role because of the signaling of the tumor microenvironment to the tumor cells. But now I just want to play these movies. I hope you guys see. This is the normal condition that these cancer cells are crowding out or invading but now this is a knockdown condition, and these cells are trying to crawl out or trying to migrate. They cannot migrate. I can play it one more time. As you see, these cells are moving out, and these cells are basically inhibited. So we were able to basically inhibit the migration or invasion of tumor cells in presence of fibroblast cells by knocking down that gene. So this paper was published in Cancer Research in 2019. Now we can do so many sort of kind of things with this tumor microenvironment. Now the next question that I want to go quickly through was, on the same signaling axis, we found the gene because of biochemical signaling factors which was promoted in tumor cells. But now what is the extracellular matrix remodeling? So for that, we engineered another tumor model on chip. So this was open top now with the same basically cell type. So you have microvale-based collagen hydrogel, micro-molded, you have these circular entities, fibroblast cells. But now we collaborated with our colleagues in physics department, Dr. Robert Ross, and then they could come with their AFM probe and then now probe the matrix and see what is the stiffness of matrix because of presence of these fibroblast cells. What these images are showing that the cancer cells are basically crawling out and they're basically invading. And now, interestingly, we can look at the extracellular matrix of collagen, specifically deposition, and when we have cancer cells in monoculture versus co-culture, as you see in the dashed red box, you see excessive amount of collagen deposition. And now, interestingly, the stiffness of matrix was elevated because of the crosstalk of the tumor cells and fibroblast cells. Now, this is showing the same thing. When you have CAFs, the stiffness of matrix is significantly elevated. Now we can go actually do molecular analysis. I don't want to go through the details of this. This was published in Biomaterials in 2020, that we found PDGF-AA and PDGF-BB are the promoters, or basically associated with this signaling crosstalk between the two cell types. So this was our first technology platform because of presence of fibroblast cells that we wanted to study what promotes tumor cell invasion. Now we can actually go and then make the platform more advanced. Now we can actually add a third layer to our platform. So we have tumor on the red region and we have stroma on the green region, and now we can add a vascular region and then load that vascular region with endothelial cells. And then these are endothelial cells that cancer cells can crawl out, and then there's a signaling between endothelial cells and tumor cells that they can go invasion into the tumor and intravasation into the vascular network. So this was our breast cancer work. Now we use this platform, and this platform also was granted a patent, a US patent, and now you utilize this platform, and there was a collaborator, Dr. Mehdi from Barrows Neurological Institute in Phoenix. And then she approached me, she mentioned that, okay, you develop these tissue models on chip, and then why don't we develop brain tumor on chip using your platform technology? And I just want to make a comment here. It's an interesting story. When I came to ASU and I started developing these tissue on chip models or tissue engineered models of diseases such as cancer or other diseases such as cardiac that I'm going to show in a second. When I was presenting this to clinicians, there were a lot of skepticism on the physiological relevance of these chip technologies. And that's a true skepticism, that clinicians always ask whether these chips truly mimic the physiological relevance. So when we published our previous paper in Cancer Research, we showed the clinical relevance. That's, for instance, the same gene that we found was well correlated to the clinical databases of the patients. But now after that, when you go and present these technologies more and more and educate the clinicians on the utilities and the uniqueness of this technology in terms of a 3D microenvironment, that on the multi-well plates, you cannot have this microenvironment. The organotypic architecture, the spatial resolution, imaging, and etc. And then when they see the power of this technology, they get really, really fascinated now. So we have been approached by numerous hospitals, and like several hospitals in Phoenix. BNI was one of them, as I mentioned, that they wanted to develop brain tumor on chip. Another investigator wanted to develop prostate tumor on chip. And then now we have another investigator from Phoenix Children's Hospital that they want to develop congenital tumors, basically. So that's our job as bioengineers, to kind of show the uniqueness and power of these technologies to clinicians and say their uniqueness. and then that we can utilize this to answer fundamental questions that which could be used for the process of drug discovery and then inhibition of the disease progression or manifestation in the body. But anyway, so that was a small story. But so I was approached by collaboration, and I was very fortunate, call it this way, that someone emailed me from BNI and said, okay, so why don't we develop this for brain cancer? And it has been a very fantastic collaboration that we have had, and secured multiple grants on this. So on this chip technology, as you see on this basically coin, as you see on the bottom photo, and this is a tri-layer. So we have glioma stem cells at the core, and then we have stroma on the center, and then we have endothelial cells. But the interesting thing is about brain cancer is like brain tumor cells are embedded in or surrounded by different niches in the brain, in the tumor, in the brain microenvironments. And so the context of different niche in the brain cancer is kind of a well-known context. So we have one of the niches which is really, really important, which promotes the brain tumor invasion, is the perivascular niche. And basically the signaling between these brain tumors and the vascular network or capillaries into the brain lead to the invasion of these cells and then metastasis. And this is one of the aggressive types and lethal type of the cancers, basically, in patients. And the other niche that has been famous is the hypoxia niche. And there are hypoxic regions in the brain that promote the invasion of these brain tumor cells. But anyway, so one of the niches that we were interested in studying was, and the signaling, to investigate what is the signaling, and was a perivascular niche. So that's why our tumor model was well suited for this study. So we developed on the third layer of the tumor these endothelial cell network first, and as you see, these are the nice formation of lumens on the outer layer. So we let the 3 days, we let the chip for the formation of vascular network for 3 days. And then we actually added glioma stem cells after 3 days. So what you see, these are GSCs. This is a 3D high-resolution confocal image of showing these GSCs, glioma stem cells, that are extending their processes toward the endothelial cells or vascular cells because of the signaling of these two cell types. And then now we have high-resolution that this movie, I don't know, is it playing, this movie?
Yes, yes.
Great. Because I don't see you guys, I just want to ask. But yeah, this is showing actually that we are getting into the lumen of this vascular network, and then these cells are extending their processes. Now, we wanted to ask these fundamental questions: that what is the signaling factor between the two cells? So we tested the invasion of these tumor cells toward the vascular network in 3 different media: neural stem cell media, neural stem cells plus serum, and then EGM2, which is the endothelial cell media. But when we supplement this chip with EGM2, which is endothelial cell media, it has a lot of growth factors in it. And then when we have neural stem cells, so we wanted to kind of dissect the role of just endothelial cells. But when we showed that when we have neural stem cells on this panel B, as you see, even just the presence of endothelial cells without any growth factor or without any specific media promotes the invasion of these GSCs, that shows the crosstalk between these two cell types. Now, we did a lot of biology on this, and we are still doing a lot of single cell RNA sequencing studies. And we have found novel factors and compounds which promote this invasion. Now, we actually added another layer of complexity to our model. So we had monoculture, co-culture with endothelial cells. Now we can add another cell type in the tumor microenvironment. It could be microglia cells, or it could be astrocytes. Now here we have astrocytes, and as you see on panel B, we show the promotion of the invasion of the tumor cells, or GSCs, glioma stem cells, toward the vascular region when you have multiple, basically, cell types in the tumor microenvironment. Now we can actually have different levels of oxygen, 2% oxygen in monoculture and triculture, and then see basically how these cells invade. Just briefly to show you the relevance to animal models, and then on the top we have PDX. So these are fluorescently tagged GB3 cells, which is a subcategory of GSC cells, which are orthotopically transplanted in mice. And then this was done by our collaborators in BNI. And then it shows that these red cells, these are GSCs, are going in a chain-like kind of migration toward the blood vessels in the brain of a mice, which was well correlated to the bottom image, which is our microfluidic platform. So this was another level of correlation that we did, comparing side by side of the outcome for microfluidic platform to the animal models or PDX models that we had. So this is our cancer work that I wanted to share. I think it has been 40 minutes now. I have about 5 to 6 minutes, I believe, to wrap up. So I'll show you a couple of other small projects, I mean, not small, other projects apart from cancer that we are working on disease modeling. So the other project that we have been working on is an heart on a chip. So this was developed by one of our fantastic PhD students who's going to graduate in the next couple of months. So as you know, in the area of cardiac, the cells, the cardiomyocytes are highly organized, and there is a structural function relationship. And this organization of these cardiomyocytes and fibroblast cells promote the function of the tissue. So we wanted to mimic this architecture here, but now we wanted to go one step further and mimic a heart attack condition by putting this chip under hypoxic condition. So this was developed actually in our lab. So what you see, actually, this is our microfluidic chip. I don't know if I have one of those chips here. I don't have it here, unfortunately. But so this is the same size that we have developed our previous chip in the size of a penny or dime. And then these are basically nice styration of cardiomyocyte, which is one of the critical importance to make sure that when you develop these chip, that these cells are highly organized. These are bottom row are showing organization of the cells and then beating signals. And then we have done different alignment analysis of these cells to show the physiological relevance in the structure. But this movie that it's playing, it's showing the cardiomyocytes are nicely beating inside this platform. Now, on the next step, we actually can now mimic heart attack on our chip. We can take our chip. So this is our chip, as you see on the panel B. We can have iPSC derived cardiomyocyte. These are human-induced pluripotent stem cell cardiomyocyte. We can differentiate them to cardiomyocytes and then purify them. So we go through 24-day of differentiation process, which is a very arduous process. I have been involved with that in the lab with my students, and I know it's a lengthy process to drive these cells and then purify them. And then now we mix them with human cardiac fibroblast cells. These are isogenic cells, meaning that the fibroblast cells are also derived from the same iPSC lines. And then we mix them in different ratios, but the 4:1 ratio, which was the best ratio, which worked for us, into the chip. And we have highly organized kind of architecture, as you see on this web kind of like feature that it's formed, the 3D tissue formed on the chip on panel C, and on panel D, we see the sarcomere styration. And then now we can put the chip under different oxygen levels. So we have 21% oxygen, which is called hyperoxia. Physioxia is just 5% oxygen, physiological oxygen level, and then hypoxia is 1% oxygen level. So we have done so many studies on this, and then we are digging into molecular kind of signaling pathways or crosstalks, and then to see what gets upregulated when you have these hypoxic conditions to mimic a heart attack on a chip. So we have these beating signals, spontaneous, and different oxygen condition. What you see on the hypoxia on panel A, on this green bar, which becomes very irregular. And then, so if you see a beating variability on panel C, when we have, just compare physioxia and hypoxia, you see that when you have a hypoxic condition, there is a significant beating variability. And also we have upregulation of hypoxia response genes such as VEGF-A, vascular endothelial growth factor, and also contractile basically gene expressions are significantly downregulated in the hypoxia condition. So with this, we are doing actually RNA sequencing again. So we have seen different pathways. HIF1 signaling pathway gets upregulated. I don't want to get into the details. Or glycolysis pathway is upregulated. And so a lot of other pathways which are relevant to cells. For instance, cell cycle is downregulated and the DNA replication is downregulated because of this hypoxic condition. So now we are continuing on this work. And then we have developed, as I mentioned, we are working toward development of other chip technologies in our lab, apart from cancer and cardiac. And that depends on basically on the clinical need and then collaborations. But I hope that I showed you the importance and the power of this technology. And then hopefully in the future, we will see more emergence of different technologies. But with this, I would like to acknowledge my lab and then current lab and then alumni members and the alumni members, and also funding sources. But hopefully, this is what I want to mention. Hopefully, in the future, by emergence of these different technologies with different groups, so we are gonna basically see multiple specific checkpoints on the physiological relevance of these chips and create a standard that when different chips are coming to the market or coming to the studies, we can see that they have met these specific standards or defined standards that they can move one step forward for reliable preclinical testing or testing of efficacy of numerous drug compounds. So with this, thank you very much for your attention. And then if there's any question, I'll be happy to answer.
All right. Thank you so much, Professor Nikkhah, for this excellent presentation and your inspiring career journey that you had. It's my really great pleasure to host you today, and also, know you from your Harvard time, and visit the lab and get to know you, and actually really learned a lot of things from you. Once you're taking a small breath, I just wanna. Can you stop your screen so I can share?
I stopped it.
Thank you so much. I just want to announce that next week we have Professor Ritu Raman from Massachusetts Institute of Technology, that she will be talking about neuromuscular actuators for medicine and machines. Please follow us on Twitter or LinkedIn to get the most updated information about our upcoming seminars. And last but not least, I would like to thank our sponsor, Montreal Transmedtech Institute, which sponsored this e-seminar series. By that, I go right away to the chat box, to the Q&A box, and encourage the audience to ask their questions. And we have a couple of questions in the box. Sure. I start with Mariam's question.
I hope I finished on time to have enough basic time for Q&A.
No worries. If you have extra time, if you don't have any commitment.
No, I have extra time, but I just meant like your timeline, but just.
It's fine. It's fine. So Mariam is, first of all, thanking you about your interesting talk. And she's asking, what are the challenges working by this chip with post at channels interface? Did you work with hydrogels? Did you see any leaking or leakage between channels? How did you solve this problem? Did you use any coating on PDMS? There are many questions, like technical questions.
Yeah, but this is a very excellent question. I don't know if the student or post scholar I would call, I don't know if the student postdoc faculty asked this question is working. Actually, it's very, it has challenges. So I had one student who was excellent in working with these chips, and he generated all those data. The next student came, he couldn't work with that because he had a lot of leakage. So here's what's happening. So I would call the challenges are at two ends. One is basically the handling of the chip, because these chips are very small, as you see, and I have worked with this chip in the lab, so I had to go to the lab and ask the students to train me. So you see that you hold the pipette, and this is the smallness of, and your eye really you cannot see the tumor region, how much you have pushed. So the hydrogel that we use is matrigel. So if you push the pipette a little bit more, it's gonna leak through the post. If you push a little bit less, you're not gonna reach the post. Now we coated the chips, yes. We coated the chips, some chips yes, some chips no. On our cardiac chip, we didn't have any coating, so we wanted tissue to be suspended. But on cancer chip, in order to attach the collagen into the surface of PDMS, so we had to go through basically SAMs, like specifically we had to add PDL and glutaraldehyde. That this glutaraldehyde actually binds to the amine group of this collagen, and collagen kind of sticks to the chip. And so now to answer your question, we had multiple challenges. A couple of students got frustrated, and it's a technology. So now we redesigned the chip a little bit to bring the posts a little bit close to each other, to have more user-friendly injection channels. So I have a master's student right now who's working his thesis is focused on this to optimize this chip, to make it more user-friendly. So because a lot of investigators we are actually shipping this chip to some investigators on East Coast that they want to use it for different types of disease, not even cancer. And then now, how user-friendly is this chip for the end user to be able to inject? So we have to fix that problem here. But I would say, if I answered your question, I don't know if I have any at two ends, but definitely there are things that you can perform on the technology itself and on surface coating to kind of reduce that manpower error. That's what I would call.
Thank you so much. The next question is from Himalata. Is it possible to develop osteomass on chip?
I am not. I am not familiar with that area, but I have actually seen that a lot of chips developed for bone and also like metastasis of cancer into the bone region, and like I don't know if that's the question, but what you mean by osteom.
Yeah, so he can or she can clarify the question if on the.
But that's not my specific expertise, because one issue when you develop these chip technologies is like the biology. We can, as I mentioned, fabricating these microfluidic chips is like easy. We can teach high school students to come and fabricate the chip, right? Working with the chip is another level of complexity. But again, above that complexity, if you just present this chip and say, I developed this technology, people are going to say, okay, it's fairly simple. But what is important is the interface of biology. That there are a lot of diseases that they have different biological, for instance, cascades that we have to understand. So that's why at the end of the day, you have to limit yourself to few number of diseases. You cannot be able to study all the diseases. Bone tumor on chip, yes, that's a question. I think that, so if I understood kind of correct. So there are studies by Roger Cam at MIT that he actually studies breast cancer metastasis into the bone. So they had channels between the channel of microfluidics. They have a hydrogel, so they load the hydrogel with tumor cells on the outer layer, so they have bone cells, and then you have to promote the invasion. So I have seen people are developing.
Himalata, yes, there are people working, and so you can go and check the papers. We have our colleague, Dr. Saudadman from Sharif University, also wrote in the chat box that they are working on the similar project on chip, and they're doing co-culture and cell differentiation. And one of our previous great speaker, Professor Kamachava from University of Toronto, also asked, first of all, thank you for your great presentation, nice presentation.
Thank you.
She's asking that I noticed that you are using matrigel collagen mixture. Aren't you concerned that you're washing away this physical gel by liquid flow?
Very good question. No, actually, I think that, first of all, thank you for attending this talk. So the Tumor is matrigel collagen, 1-to-1 ratio. And we tested different gels to see which one works for the two. So the way it works, and the reason that these chips were granted, like basically, I think the patent, because the examiner was asking us, what is the novelty of these chips compared to the previous chips? If you look at the literature, a lot of chips, most of the chips, they have Y-shaped kind of architecture, meaning that the gel is at the center, is surrounded by two media channels. So that both sides of the gel are basically exposed to the shear stress. Here, our tumor is separated. So we have the tumor, inject the tumor first with collagen matrigel and cross-link it. And then multiple inversion to have cell distribution. Then we go to the next step, and then basically we cross-link on the stroma. Then we add our media at the end, after cross-linking of the gel. So the gel is already cross-linked when we add our media to the outer layer, which is exposed to the stroma. So we have never seen like being washed away by the media.
Thank you so much.
That's a very good question.
Thank you so much. There is another question from Londano, my colleague in Montreal Transmedtech. First of all, she's saying that the awesome technology to study interaction and physiology of different cellular types in the system. Then she's asking that, can you comment on the advantage of working on chip instead of the classical cell culture using mixed cultures? Is it related to the organization in the chip or only to the scale? Thank you.
I think it's related to both, but the main thing that we were looking at, that's why I had the word in the title of one of the three papers that we published. We specifically designated the word organotypic, and then so what we mean by organotypic is that we have spatial organization of the cells. So we can have the tumor cells at the initial architecture separated from the stromal cells, but let them intermingle later as they invade. But when you have standard even in a lot of microfluidic devices, the same Y-shaped channel that I mentioned, there are not different compartments for different cells. So you have to mix all the cells together and inject into the tissue region. But for us, it's a multiple, basically, steps that we can separate these cells. That gives a nice spatial organization and organotypic architecture. But in the multi-well place, you have to mix the cells. There's no way, unless you use microcontact printing, that you print one cell type and then you print the other cell type, but they are still on a 2-dimension. They're not on 3-dimension. And also in terms of scale, we use a few thousand cells inside our chip. So it's a very, very small scale compared to the multi-well place that you have to use a lot of cells, a lot of media, a lot of antibodies. But yet we use significantly less amount of reagents in the cell, but the assay is compatible with the standard like biological things such as RNA sequencing, qPCR. The only thing which has been challenging is the Western blotting because of the amount of protein extract. But I hope I answered your question. But our first thought was the organotypic architecture and organization. That's what I would say.
Thank you so much. There's a follow-up question from Professor Komechawa. Is cross-linking explained enough in your paper?
Yeah, I can send the paper actually if I have the email. There are two papers. The first paper I showed today was published in Scientific Reports. So that was basically to show the validity of technology itself. We have explained the cross-linking. On the second paper, which was cancer research, which is more biology, we have also explained. So most of our papers we have explained, but on the one comment I just wanted to mention also, as I mentioned, the uniqueness of platform is we can use different gels for different regions. For instance, for our brain tumor on chip, we use fibrin for the outer layer to promote vascularization. We didn't use matrigel, but it was cross-linked nicely. We mixed fibrinogen, thrombin at different ratios, and then we cross-linked it, then we injected matrigel into our chip.
I can put you in contact and share the Professor Komechawa email with you, so you can send the email. And by the way, I want to say hi to my classmate, Professor Saodat Man from Sharif University, that she's here with us today, and she's working on the same field. So there are other two questions in addition to my questions also. From Nike, is there any possibility of contamination of the microfluidic system? Does the chip require a specific temperature or condition for long time storage?
The chip itself, that of course contamination is something I cannot separate something called the chip contamination from general contamination. So we might have general contamination in the incubator no matter what, when you're working with the cells, right, or media. But the chip itself, we actually after we fabricate the chip and we surface treat the chip, we pass before surface treat, we pass the chip through cycles of autoclaving. So that autoclaving cycle actually sterilizes the chip. And after that, all the steps are being performed under biosafety cabinet in a sterilized environment. So that's how we can reduce the contamination of the chip. Knock on the wood, we have not knock on the wood. I mean, 7 years, we have had maybe one, not for our chip, for our general cell culture, we had just one but for our chip, I've never seen a contamination issue. Like when you go through those steps, these are well-known steps, it shouldn't be a problem.
Thank you so much. The last question from Nahid is thanking you for the interesting topic. Is it possible to mimic circulating tumor cell adhesion and extravasation by chip technology?
Yeah, extravasation.
Sorry.
Yeah, no worries. No, I think so. There are two factors that you have to consider here. When you want to study extravasation or intravasation, the endothelial network formed in your chip could be two ways to form the endothelial networks. The way that we formed endothelial networks was like by mixing endothelial cells into a fibrin gel and injecting into the outer layer or the third layer of the chip and let the endothelial cells spontaneously assemble into a vascular network or capillary-like networks. So over there, we can have circulating tumor cells, but we have not studied whether those capillaries, with the size of those capillaries, is enough to study extravasation. So the best way to study extravasation, and we developed another chip for that, is to use a wide channel of 100 micron and seed endothelial cells on the side of the chip, and then let the cells, endothelial cells, attach to the chip, and then pass your cancer cells and then see whether they extravasate. And there are numerous work by investigators such as, I think I don't remember the name, but there are several papers that they have worked on extravasation. We work specifically on intravasation, meaning like from the tumor to endothelial network, and we showed that the tumor cells, they cross the gel and then they cross the barrier of endothelium and get into the network. If you saw on the slide I showed on the tri-layer, and there were red cells into the green kind of capillary networks of endothelial cells. We didn't specifically study extravasation in our chip. It would be a little bit challenging for us because our chip is curved. So getting endothelial cells on that outer layer might be seeding, might be a little bit challenging. So we should have a straight channel. But a lot of people have studied that, but definitely there are capabilities.
Thank you so much. Now I switch the gear to the cardiovascular section of your talk.
I was thinking of, because of time, I was going a little bit fast because I didn't want to get the presentation too long, but just show
No problem. Usually we stay with the audience if they have question, and if the speaker also has time,
I have
a little bit more. So, I mean, do not hesitate if you are committed or something.
I have more than enough time.
Okay. So my question is that basically, when we write grant on heart on chip or other, cardiovascular on chip or heart attack on chip, as you presented, reviewers ask this question that, okay, there are many heart on chip in the market or developed before, and what's actually your novelty, and without systemic effect or systemic interaction with other organs, or including the immune system into the system. And so I always ask this question from friends and colleagues who works on cardiovascular tissue engineering. What would be your perspective or your answer to this question? Because the question that you're asking is about cardiovascular diseases and the stuff, but they're asking you to add multiple organs together to see the systemic effect and also to see the immune system response. So how do you answer this question to the reviewers?
Just to clarify, multiple organ, you mean like multiple, basically like liver, cardiac? Is that
downstream and upstream, like the systemic interactions.
Sure. This is first of all, it's a valid question. I'm not claiming here that this is the best chip or this is. I think these are bold claims that we have to be cautious about it. Like when we talk about it, and we have to, honestly speaking, we have to maintain a neutral, basically language. Like there are definite advantages on other chips, and some advantages on our chip that we have to. That hopefully, as I mentioned, at some point the community is going to come together and basically present some standard points of these chip technologies and enrich the knowledge. So in terms of novelties, valid of multi-organ immune cells, I will mention in a second. In terms of our chip specifically for our chip technology. For our chip, there was no microfluidic device on a small scale. It would lead to highly organized fibers of cardiomyocytes embedded in a gel in a 3 dimension with cardiac fibroblast cells. So that was the novelty. That, yes, there are for instance, the work by Dr. Gordana Novakovic or Melissa Radzev, they have mostly, or the work, those are excellent work. They're published in great papers. No question on that. But just Mostly they're like two posts, and then the cells are embedded in a fiber, and they're like basically beaten. The unique aspect of our technology, we wanted to introduce that alignment into a microfluidic chip, which is surrounded by two perfusable channels that we can see the diffusion of drugs. So we can control the diffusion. Because on those assays, so the media is on top. You see my point. So the uniqueness of one. There are chips or there are tissues, cardiac tissues, that they have microgrooves. They are highly organized. They're microposts, but they're not in microfluidic chip. So we wanted to bring these together in a design of a single package microfluidic device that you have highly organized 3D tissue in a chip surrounded by media channels. So that was what we wanted to do, was add one step to the community and say, okay, there's a chip. But complexity. It took us about a year and a half to optimize the dimensions of the chip, because it's not a simple chip. Like cancer chips, cancer cells, apart from tumor and stroma that they're in a semicircular fashion, you mix the cells with gel and you just inject. But here you should make sure that the cells are highly organized inside your 3D tissue region. So the microposts that I showed you, for instance, elliptical microposts, what should be the dimension of those microposts? What should be the spacing of the microposts? We characterize the chip, interestingly, with rat-derived cardiomyocytes, but believe it or not, when we move to stem cells, human-derived iPSCs, they behave differently. So we had to go back and look at some of our factors. So optimization of that took a lot of time. And also what ratios, that one was optimized. The second one was the ratios. And I'm getting to your question by explaining these steps. Ratios of cardiac fibroblasts to cardiomyocytes that induce to a good beating behavior, or like physiological beats. What should be that? So we tested 1:8, 1:4, 1:2. We cannot have too many cardiac fibroblasts or too little cardiac fibroblasts. And so when you see these complexities, then basically adding immune cells on top, which is a very valid comment, especially for heart attack. We have just co-culture fibroblast cells with cardiomyocytes, but now we have to add, so we want to go in a stepwise and methodological fashion to kind of add complexity. If you want to add all complexities all in once into the chip, you have not yet optimized your geometry. You have not yet. But our chip is the next step right now to go to the next, for instance, add immune cells. So that's the first question. But I think for us, this is a thing that I'm talking about myself and my own group, my work, my students' work. They had to go step by step. So I couldn't ask them to add immune cells, fibroblasts. So that's the first thing. And multi-level organ and all the diffusions and the crosstalk, it's another level of complexity that we have not developed that in our lab. But these are very valid comments. And then just to let you know, the last comment is like the cardiac on chip and all these organ on chip, even tumor on chip, is a very, I would call it very competitive. Like there's several groups developing these things. But these are definitely valid questions, you know. But you have to find your niche, and where is the niche?
I think the reviewer didn't want to give us the grant.
Exactly. That's
I
think it's a learning curve. When you submit the grants, and you realize which, like, let's say if you want to write on just heart on a chip, I think because there's so many platforms are out there, unless you have a specific biological hypothesis or a biological question that you want to answer using your technology, then that brings novelty. If there is a disease, for instance, one of our projects right now we are working is on LQTS2, a long Q syndrome, right? And then we use we edited the cardiomyocyte with CRISPR-Cas9 to induce basically mutation. And then now we are adding that to the chip, which has a different novelty. So it's basically on a microfluidic chip. But so that depends on question. I agree with you. It's very competitive. I think it's.
Yeah. Anyway, I remember that you told me a couple of years ago that cardiovascular tissue engineering is a competitive field. So
I think everything these days in bioengineering, everything like tumor on chip, cardiovascular on chip, like you see the number of papers are coming.
Exactly. Wahid asked a question that he's thanking you about for your fantastic works and information about your project. As you mentioned, it's so hard to provide proper cells from patients, and one of the ways to be cost and time effective is using simulation software such as COMSOL, as your team worked with it too. Is it possible to develop a more precise software model based on the results of your work to use in the future investigations?
I think COMSOL we used for diffusion of molecules into the tumor and stroma in terms of fluid. I would think like in terms of fluid dynamics or fluid mechanics of what's happening with the flow, shear force, velocity, diffusion. Those are the things that you can predict with COMSOL, and you can definitely. But in terms of cell behavior, I'm not sure. Is that a question? I don't think that's not the COMSOL is intended to be used for. We can definitely do something. We can definitely take these bidding patterns or basically patterns of cell invasion from the chip and go back and create a large data set and now use machine learning algorithms for basic. So that's what people are trying to go based on, like, you have to develop multiple kind of layers of data and then train your basically algorithm, and then now you can have predictions, right? So, but that's different from COMSOL, I would say. COMSOL, what we use is mainly for what's happening in terms of fluid dynamics or fluid mechanics inside the chip, and these are like laminar flows, so it's very simple to study.
Thank you so much. One question I have is that in microfluidic, we usually try to scale down to micro and nano level, or nanofluidic even. So to save media, cells, and everything. So, but is there any need to scale up, for example, to have a solid tumor?
I mean, the size is, that's a very good question, but you are limited by diffusion.
If it is vascularized, I mean, if you have a vascularized tissue.
If it is perfused, then you can actually, but provided that you can control the microenvironment. The reason that we scale down, apart from cost, apart from amount of, for instance, reagents that you want to use, and apart from the control. But the question comes into the fact that, like, even for instance, for a tumor, if we wanted to double the size of a tumor, now we have to be worried about formation of hypoxic core. Now, how do we address that? Then you have to induce a vascular network. How does that going to assemble into your tumor, lead to the perfusion and those kind of things? I think it's a good question, but I think those are the things that we have to perhaps. One thing we can think of, and we started thinking of, I don't know if I presented that in your class, Human, or not, like a high throughput microfluidics, like basically you can arrange, but that's scaling up in terms of the number of platforms that you can actually have rather than just the tumor itself, But that's something which has been our concern, that like, and also if you enlarge the tumor, when you have the media, how much when you want to stain, for instance, using antibodies, what is the distance limitation that the antibody can penetrate through your tissue? Those are the things. So one of the colleagues actually developed a technique using a vacuum for a deep tissue staining. Those are the challenges come into the front. But my main concern would be like cell death and hypoxia. We have one project in the lab that we created cardiac organoids. Those are scaffold-free cardiac organoids that could be injectable. But someone brought these size is 150 to 200 micron, but then it's very small still, like it's a bit in the diffusion, but like we had questions that do you have hypoxic core at the center of your tissue?
No, for sure you need vascularization.
Yeah.
So it's questions are coming. So thanks for your interesting talk. This is Ali is asking, is there any possibility that the chip size and volume can affect the cancer cell secretion of the cytokines and derive phenotypes? Cancer cells in the body have an open source to what is needed for their aggressive mutations other than what we offer using the chip, which can be in a way or another 100% accurate. So it means that I think this is along with my question, when we are scaling down or up.
Scaling up. I think in that scale, I don't think like that the flow on that scale, if you go by 100 microns here and there is going to influence the cell itself, let's say, unless you, by scaling up, you create a hypoxic core, then now they start secreting VEGF, vascular endothelial growth factor, right? And then so that happens. Actually, what we showed in our heart on chip, when you put the chip under hypoxia, the VEGF secretion goes higher. So that could be the consequence of that. But so we have to be cautious about that, I would say, and then see what really we are looking at. But definitely, if you manipulate the environment one way here or there, you're going to influence the cells.
That's true. That's true. Because we had one of our presenter or speaker was Professor Peter Zandros from UBC now, and he was talking about solid tumor and like a centimeter scale tumor, and he was talking that there is a big need for that. So in terms of studying, like the cancer microenvironment or environment in a more clinically relevant, structure.
But definitely that requires, I would think of like we have to go study the first on that. If I want to recreate that, I would go and do study like what is the vascular there was a study, actually, that's a good question. Good point. There was a study that they looked at the tumor itself, and then they created a template of microfluidic out of that tumor and like irregular vascularized network and those kinds.
Like a topography, like a stamping.
Let's say our vascular network is in a curved kind of fashion, in kind of like organ. For them, they learned the organization of vascular networks directly from the tumor tissue that they sliced, and they copied that, created the PDMS mask, and then they replicated that. It was very interesting.
Mostly it's no vascularization, so it's going to be very small vasculatures.
Capillaries, yes, on that scale that they penetrate into the tumor. Of course, it's good if you have a technology to kind of like develop that, but I think it's something that, but I think the work of I would say work of Stephen George, he's one of also great investigators. I don't know if you know him or not.
Yes.
UC Davis. He's actually, he developed this kind of like elliptical shape tumor, which is penetrated by vascular networks, and they mix cancer cells into that so they can look at intravasation extra. So I would call it, that's a larger tumor size, which is already has a vascular network in it, and it's connected to the outer vascular network. Larger kind of like basically capillaries. So that's a very interesting setup if someone wants to look at, someone wants to address that question, which came.
I will check.
By Dr. Steve George, actually, in UC Davis. So his tumor model is a very interesting model too.
I will check on. So the last question before we close this session is from Ermia. Thank you for your great presentation. Regarding the recent question, do you think machine learning could be employed to have a better estimation in compared to COMSOL?
I don't know. I think machine learning, but people are using it for, I see that it comes into the field of biology right now. But COMSOL, as I mentioned, we use it for different applications, like diffusion, COMSOL is a like multi-physics, basically. What is happening in the physics of environment, like in terms of like diffusion of molecules, fluid mechanics, shear stress, and those kind of things. That's different from what I meant by machine learning in terms of like cell behavior. Like what are the learnings from these kind of like compounding factors in there in terms of biology. So I would call it a physics and a biology approach. That these are the two approaches. Like COMSOL is used for the study like physical phenomena in the microenvironment rather than biological phenomena.
Thank you so much for your time, Professor Nikkhah. I hope that we could host you in person in Montreal soon. And then we can chat, we can actually discuss this kind of projects and collaborations with each other. And I would like to thank the audience also for asking many good questions today. And just remind everybody that we continue these seminars next week with Professor Ritu from MIT that she's talking about neurovascular actuators. So by that, I would like to thank everybody. Take care of yourself and see you next Wednesday at noon Eastern time.
Thank you very much. Thanks for your kind invitation. Thank you. Thank you.
Take care. Bye-bye.
Take care. Bye.